Hello students, welcome to EPG Pathshala. I am Indira Ghosh from JNU. Today we are going to talk on the module which gets Proteomics, Transcriptomics, Metabolomics in biology under the paper of Computational Biology. Now, plenty other omics are there. However, I will try to address within small scale transcriptomics, proteomics and metabolomics data integration, particularly in the context of disease as well the plants under stress, how we can understand both the context. Well, I would like to start with that the context is disease or complex diseases and how we use the transcriptomics, metabolomics and all other omics into finding recovery from these diseases. So, the basic motivation of connecting and integrating all the omics is the genetic factor and environmental factor do a balancing act on the species of any genome or any proteome, metabolome, etc. And if we, the disease caused because we have an abnormal state, which is normally it is a homeostatic standard mechanism of a living body. So this disease can be, as I said, that can be caused because of genetic or because of external things like pathogen, chemical, lifestyle, etc. And major genetic factors which involved in complex diseases, they act by a function through the complex mechanism. And this function are interacting between many of the omics data. And in most often, it is the gene product which is changed. So, trying to give the type of biological interactions, we can look into like an entities as a computational biologist would look. And these entities can be, if we take the previously shown one slide, which I have detailed in earlier interactions, how the entities which are biological entities like the motives like gene, DNA, protein, etc., molecules or the motifs or the pathways, etc., and how they are influenced or what are their biological malfunctioning or functioning. So on the left hand side I showed you disease can be gene disease association, then functional motifs can have gene and functional association. There could be pathways, which of course regulation of the metabolic pathways, and at the macromolecular level and molecular level, that are gene, protein, metabolic, mRNA, etc. And their interaction between proteins or DNA protein and sometimes co-expression, etc. can influence and get you the disease. So we will try to address few of these. This is last 15 years or so has come up and aid the research in biological and disease associated science called GWAS. This is nothing but the genome-wide associations of the sequences. And this has come up with lot of people, that is human, who are patients, and there are plenty of people who are not patients. So if we compare their phenotypic, that is simple high diabetic or not, or blood count, etc., and we compare with the genetic marker like SNPs or single nucleotide polymorphism across the entire genome, we might be able to compare this. Now, there are two major technology which has fed all these data generation. One is microarray chips, which are also shown here, which is the expression of how much expressed RNA can be identified and compare them with the normal ones or not having the disease ones. Another is a chip technology, which is called chromatin immunoprecipitation. That is another technique which has been used. Many cases, chip technology is followed by microarray technology, which is called chip technology, or sometimes chip technology followed by sequencing has become mostly very widely used to find this in vivo mapping of human patients. This can be extrapolated to the farm animals and also has been extrapolated in the plant biology. The limitation of these association studies are these variants are identified may or may not have in reality functional consequences. Many of them just been probably mutant but not functional. May not be right now, may be just done. And then sometimes it may be in the genomic region or maybe in non-genomic region in other places. But they might have influences. Then not many markers are there. If you have a huge population, then statistical test increases the likelihood of false positive if we have not used enough number of markers for a large population. Also, there are difficulties of producibility sometimes, and sometimes very rare variants occurs which might be missed because and maybe ignored. So other technologies comes to help these kind of limitations. The proteomics. So how the proteomics can be? The information collected from the disease variants or genomic variants or from SNP genomics can be as well looked into at the protein expression level. So one can look at as it's shown here using mass spectrometry very well and target identification, then resistance to cancer or some populations, then find that drug action. So I am showing here blue as if it is the way of looking at whether those particular proteins are sensitive are under the protease inhibitor or under phosphatase inhibitor. Protease inhibitor would give one type of map of the picture of the proteins, and phosphatase inhibitors would give another type of map. So we can have digestion and non-digestion. We can have different kind of systems. Also, people use isotopic labeling sometimes, and all these labels can be collected as fractionalized and then look into the HPLC and MS, that is by mass spectrometry, so that one can identify these proteomes associated with patients or not associated. So these techniques are used extensively to aid the presumed earlier the SNP concepts were from the disease association with the proteins. The most difficult part here is to integrate all these information. So you get information from proteomics, you get information from microarray, that is transcriptomics, you get information from genomics. How we integrate them and what is the big difficulty? This is the question must be coming into your mind. That most important difficulty is not the format, not the large data, not handling of the data. Even it is not the curation of the data. The most difficult part is called validation and in vitro manipulation and functional understanding. So we can get different kind of data on different level of connectivity, and we need to build certain models to prove or find out whether we are right scale or not. Now, many cases, another set of variance checking is done by using drugs, antisense, and sometimes the clinical validation. However, the most difficult part is lack of coordinated effort on this proteome discovery. That is, any drug development would require a information connectivity amongst all of them. And that is where the challenge of bioinformatician comes into picture. And in future industry, all these information along with the phenomenological information or doctors checking up has to be integrated to get the new future leading towards required medical intervention. When we understand that about the disease, to include in our designing of interruption to the or health management, we require to have molecular data as well as clinical data. So we should use these data in together and use the techniques like bioinformatics tools, semantic natural language processing tools, and the statistics and data integration tools to connect the clinical application for human health to the molecular and clinical data. These are listed many of such omics which will be also utilized under the different topics of omics. However, within period of time, I would like to address only few of these omics and continue with mostly the proteomics, transcriptomics and metabolomics in my following talks. As I discussed about so many different omics, I'm just showing you few trends of data growth, and that will be enormously understand. So genomics data, and here it shows that just as you see for two plots on the top are the gene bank data, which is just the sequence data, process sequence data, and WGS, which is the GWS data, that means whole genome sequencing data. As you see, more and more, this is red color is going above the blue color, which means that we are having really huge amount of data searching. Microarray data and proteomics data also are collected, parallelly analyzed, and replaced by that RNA-seq data and proteomics data. However, metabolomics data still is growing. However, I will show you in later part how they also require the attention. Now, what we can do, why we integrate? Because here is an example how we can have relation. Like we can do a integrated and standard representation if we do it. For example, some of the database which I will show you in future too, that is like OMIM, which is connected with the Mendelian and complex disease relationship. And if we connect toxicogenomic database, which is what are the chemicals which causes toxicity and what is the concern, and this we connect with environmental mostly with the diseases, then we can actually do some kind of shown here the overlap of disease and overlap of protein numbers, which is written here. The gene numbers are written here, which produces proteins and derived from UniProt. You can see it. It's a really prioritization of genes can be find out. This is the beautiful system one can even achieve. So one can achieve these by technical terms by calling semantics method, which have been detailed here in the slides. There are semantic methods is taken in linguistic and computer language, and here it as biology, it mostly concept are ontology to derive different kind of information or text mining used for context based. One example is given here, like if you call APC, it can stand for antigen processing cell or presenting cell or argon plasma coagulation. Depending on the context, you choose the meaning of it. So you correlate. This is a simple technique. Using this, one can actually approach the semantic based system as followed. And here I show you we have developed a diabetics platform by extracting these meanings, and it is already working, and using the data, web data, mostly web data, using certain computer technologies which are written as RDA, OWL and SparkQL, one can find. And we just have connected all of them and come out with a system. Slide 14 shows the system that where we can do the query. Like we represent subjects as one of those entities, if you remember. It can be protein, DNA, etc., gene, etc. We can have a predicate that is which is connected to it by its function and the object also. Using these techniques and the languages and computer data processing and methods, I show you actually pictorially the workflow of how to do these techniques using semantics and using the IT, most of these like RDF, RCQL, etc. techniques. So basically this is and shows you the workflow where the techniques and the data input are both shown in one platform. The previous workflow is now shown how we develop the DAO and this is published papers of ours, where this complex disease such as diabetes actually has been developed a semantic web site, and given a query, one can have a list of different association methods and find the network analysis of the systems. Here what we are trying to say is integrating all the different kind of databases using the technique called semantic approach and computer tools, organizing the data and using freely to connect each of them. We have developed these for diabetic disease to identify what are the genes could be prioritized for the association. Not only that, which pathway and which macromolecules they actually intervene. I will talk about now about some important databases and their connectivity to connect them and how to get them as a platform to utilize our purpose. So now I am going to show you, when I discuss this, the database and connectivity, few databases which actually are easily connected and can be connected, and many such connectivity has developed lot of new information. One of them is a database of SNP. It is necessary to know this because I have talked enough on what is nucleotide variations and particularly single nucleotide variation. Here is the picture of the whole system and how to work out and what are the features it has. OMI, which is most useful for disease identification because it has contains many of these online Mendelian inheritance of man in man, and how it is structured and how is it, what are the data content in it, all are shown in this particular slide. Almost 1,500 over than that gene are being listed in it. This is, earlier I spoke about it, genome-wide association study, and this is a database which contains catalog almost more than 3,000 and publication derived and unique SNPs, 60,000 unique SNPs. So this is increasing order numbers and very nicely well built things. It contains different cases, it contains the controls and shows different ways of what you call volcanic plot, which is shown from the publication. Before I go to the next level of many other databases which would be useful and connectivity necessary, I would like to speak little about the gene ontology. I would request all of you to go into the paper which is cited down there and online available this thing. Go Ontology actually lists molecular function, biological process and cellular location. These three are wonderful connectivity because each one of them more or less connected, not well, but somewhere they are overlapped. However, they speak different objectives. And using gene ontology, how the gene ontology has been done is that it is contained all this information, but it is a machine legible functional annotation has been utilized, and it is like a direct acyclic graph, which I'll come to when I talk about more about the graph theories and how network systems are worked, which is in following modules. However, how the data has been done is most often by homology based transfer, that is, it's been found homologous to different places and found it, or by using some functional association matrix, and that is what is most predominantly related for gene ontology. Then there are phylogenetic consideration. Of course, there are text searching as well, co-expression, co-evaluation. Nonetheless, more important that it not only takes the sequence, it also takes structural alignment and structural pattern similarity extracted and put it in the information. So gene ontology is one of the most important resource to do research on any kind of omics. I will also separately address three different databases which will be very useful for metabolic control analysis topics, which is followed next module. One is human database, which is cited here. Next slide, which contains the KET pathway, and next slide contains MetaCyc. All these three systems actually connected by different kind of metabolite of human, bacterial and eukaryote, and sometimes even there is a plant-seq which contains the plant's equivalent pathways, which includes the GO terms, that is gene ontology terms. It includes pathways, enzymatic reaction, transport reactions, polypeptides, protein complex, enzymes, transporters, compounds, even which are metabolites, and many of them contains the tRNA or transcription units and protein connected to all the available NCPI and other EMBL connectivity as well. So these are the beauty of systems of omics study. Platform has been built up so that one can develop this interconnection as and when you would like to work on the understanding of disease. Here I show you the integration example of fewer omics. Now, this is a project supported by DBT and work between JNU and IIIT and running a nice platform for people who are working on most of the plant metabolomics. And why it is? Because it has been stated, as bottom there mentioned, that to connect between the genome and the phenome, you require to have this metabolomes as its information. So, as it showed in the triangle, one hand's informations are available, the QTL, etc. Another, the planar straight line, that is also available. The information is not available from the genome and the phenome. How to extract, connect them is the approach like what we have done here called CCPM. Here shows you why metabolomics took a center stage. Publication profile, if you look at that, it has been a very exponential growth from 2000 to 2010 and 13 almost. And the reason behind it is that it has the most vital, important tools like HPLCs, MS, etc. technology, which has fed it to become good platform for metabolomics identification in plants as well as in human. Well, as I have discussing now about the platform, what would be very interesting and in future can be utilized in plant metabolic systems, I would like to bring to your notice one of the interesting paper in 2007 where Russ Altman has talked about the bioinformatics is a study of two sets of information flow. What it means is there is an information flow which by central dogma is the DNA, which is shown on the top called based on the central dogma. But what we over the period of last 50 years we found is there is another information came up, and that is thanks to computational biologists that this is a scientific database and these two can be connected and we can integrate. The first one looks after the gene regulation and protein sequence and how they function. The second one experimentally connect the interlinking between them. Hence, attempt has been done to connect these two in future so that we can make a relation between genomics and phenomics. And that's the place, the most dominant role in the metabolomics platforms, which actually tries to make sense from genomics to phenomics. So, what is the utility? Once more to emphasize, this is developed for comprehensive study under genetic and environmental perturbation. So, what applications? In infectious disease, in cancer, in neurological disorder, environmental stress on plants to understand, many of these lipidomics and metabolic profiling for bacterial effect, clinical microbiology, every place you find metabolomics is taking the center stage. Question may come, why plant metabolomics is so important? Of course, plants to be really important in the future food and survival of almost the human and many farm and many animal survivals. But one has to look into plants are very complex entities, and their metabolomic system is even larger system. Animals have the contrast to animal system, plants have high ratio of number of diversity of metabolomes with respect to enzymes pathway. So obviously, to connect the functioning, many opportunities are available for case of plants, and under the stress we have tried to bring some beautiful systems so that we can provide the metabolomic informations, connect them to the pathways and proteomics and genomics, so that the scientists can build up new model, etc. So I will talk about those in few more slides, followed by slide which connects that development, genetic and environmental changes with the molecular mechanism and genotypic, and it should be readily accessible to the scientists for this platform. That's the objective of this particular platform was. We have actually integrated all possible ways, and this is a collaborative platform because it needs experimentalist input of their data, and it is a very nice secure repository so that they can understand the metabolomics in perspective of already known. It also have visualization and integration, and thanks to the DBT we developed that software. Contains the status of all that database content. So this is a list of, some of them I have shown you earlier, some of them what they contain, but these are almost simple list of these. Over more than 50 metabolomics database available. However, these are very standard, and I would say better to include them as our input files for understanding of the metabolomes. Now I will show you the overview of this CCPM, which I would request you to go online, available 2 website links, and log into it and do the projects. There are a lot of new projects we have done, literature review, which students can actually work out on them. So you can take those and understand them. It has 2 of that, how to log in. It has how to log in, and all the features are shown in black and red colors. Some of them are highlighted. It enumerates the features which contained under this CCPM, and data uploaded are from LCMS, GCMS and NMR, all the 3 types of things. Then pre-post process, then you can have proper correlation to find. Also it has connectivity to pathway. This shows one of the module which is called 3B module, data analysis sample plot. So all the plotting you can do by any other software, mainly background running program is R. You can visualize here, and it is shown the sample of pictures. So if you have data, you can input and actually analyze the data by this using the same platform. Most important here is to note that not only you find the important and highly available or differential plot gives you one of these metabolome, how you connect it with the path ID. Here I show you the red color things shows that that particular place, that particular 1 or 2 metabolites are increasing order, and green shows their absence. So you could see two types of colors in these plots, and which can be also, if they are not changed, even that also is shown here. And here is an example of connectivity of genomics, proteomics, transcriptomics and the pathway under the platform of metabolic analysis. Now to come to the summary, cellular system is an integrated system, and it is a modular system. Hence, it should be possible to connect this omics data, integrate it, and context should be functional relevance. And if we look into that, then and we put also which helps us to understand the disease, then it becomes very relevant for medical purpose. The techniques what you require to understand this different kind of variable data, different formats, and meaningful information to extract, you require the ontology, you require the semantic and natural processing language process, so that you can understand the biological systems in perspective of several omics data. We also have established and shown you that not only in human disease, even plants under different physiological conditions, we have developed only under the abiotic stress. However, this can be expanded to other stresses as well. And integrate and storage of data from experiments are most important in this aspect, as well providing them a utility front where they can seemingly simple way they can go from one to another type of omics data. They can load it, they can analyze it, would help us to build newer theory, newer understanding of the biology.